Good day, everyone. So for today's lecture session, we're still on the basic principles and concept of, at this time, will be immunology and part of pathology, part one. So I divided it two parts para dili kayo bugat, and you can digest the first part of this lecture session. Before we start, we're going to tackle on host specificity. So here, the traditional view of host-parasite interaction asserts that a symbiont becomes progressively more specialized through evolution, which means that the increasingly limits the potential number of host species that it can affect. So it increases host specificity. So host specificity means that the parasite lives all throughout its life cycle in just one host. And then compared to the general species, general parasite species, that they will be dispersed unequally among individual hosts from several different species. So, diba, na certain parasites the need of different types of hosts for their life cycle. So, also host specificity influence its behavior and ecology of both parasite and its host. So, um, so the range and diversity of host species that a parasite is capable of infecting considered a crucial measure of parasite's potential to shift host and trigger disease emergence. So here, susceptibility and resistance. So we know that the host is susceptible when a parasite, when the host cannot eliminate the parasite before the parasite can become established. So here, by definition, susceptibility is the condition of the body that increases the likelihood that the individual will develop a particular disease. On the other hand, resistance is the capacity of bacteria to withstand the effects of a harmful chemical agents. So here, the host is resistant if its physiological status prevents the establishment and survival of the parasite. So here comes the immunity. So the immunity, by definition, the capability of multicellular organism to resist harmful microorganisms. So there are three types of immunity. This is innate, adaptive, and passive. So as we say innate is it's a general type of protection. We all have it na. So for example, the skin acts as a barrier to block germs from entering the body, and the immune system recognizes when certain invaders are foreign and could be dangerous. So second, adaptive is this type of immunity develops throughout our lives. For example, is if we are exposed in a certain disease or when we're immunized against them with vaccine, so we're adapted with a certain immunity so that when a particular foreign material in fact will infect us, maka-fight ra ang atong immune system since we already acquired the immunity. Next is passive immunity. So this is borrowed from another source and it lasts for a short time. So example ani is the antibodies in a breast milk gave a baby temporary immunity to disease the mother has been exposed to. So mo nang gingan gid nila nga breast milk is important for babies kid up to 2 years of age. Since ang bata is still developing its immune system, mo nang mo rely gid siya sa iyang mother's milk since it helps the baby as well nga ma-immune siya or maka-help siya nga maka-build up sa iyang immune system pod. Okay, so we will be focusing on two, innate and adaptive. So cells in both the innate and adaptive response detect many molecules in their environment that bind to receptors on their surface. So this results in initiation of intercellular signal cascades. So this is basically cellular biology. So I hope you already taken cellular biology since I think parasitology is very specific. Maganda siya nga type of science compared sa I think dapat i-take una ang cell bio prior ani. So okay, so innate immunity, so this mechanism defense does not depend to prior exposure to invade. Unlike adaptive, kailangan pa nato my experience prior to have the adaptive immunity. So unlike siguro sa COVID, we did vaccines to have this adaptive immunity just in case COVID-19 virus will infect us. So man siya, that would be adaptive immunity. Somewhat shortcut kaysa maghulat kang acquire nimo and then you have no chance or little probability of surviving. So motong vaccine is the shortcut in acquiring adaptive immunity. So these are the abbreviations and acronyms in chapter 3. So more or less lang man sad ang needed ani or ipakita. And so, and then, so dili kay ni siya, only few naman po ako magamit ng abbreviations. But moving on sa part 2, probably na sad. So we will know. So moving on sa atong innate defense mechanism. So this type of mechanism has two types of markers, which is katulad self and non-self markers. So here, the unbroken surface of most animals provides a barrier to invading organisms. So this is useful functioning of the immune system of defense requires distinction between and cells in an animal's own body. Self, so more recognized nga, part of the human host and also are part of the human host or host. And those of another individual or foreign materials are non-self. So they recognize it as a foreign material. So here, an illustration between identifying self and identifying non-self. So moving on, so we're focusing on the self part. So all nucleated cells in the body possess unique and distinctive service molecules which identify as self. So under adding a self marker would be the MHC or the major histocompatibility complex molecule. So they functions as identification tags. So this immune system will not normally react to cells bearing these genetically determined markers or self-tolerance. So most likely this type of MHC, there are two types one. So say MHC1 are cell surface recognition elements expressed on virtually all somatic cells. So these molecules are simple sample peptides generated within the cell and the signal the cell physiological state to effector cells of the immune system, both T lymphocyte and natural killers. So MH1 molecules are cell surface recognition elements. So here, there are two types, class 1 and class 2. So class 1 proteins are found on the surface of virtually all cells in a vertebrate, and class 2 is found in participating in the immune response, such as lymphocytes and macrophage. Next is the non-self. This is any substance that is recognized as foreign and capable of triggering an immune response. So here, antigen is involved since that they are recognized by lymphocytes which bind to and detect the character shape of an exposed version. So exposed version is epitope. So makita sa ninyo later on sa akong pasabutan ng epitope during the overview of a cellular signaling cascade during immunity. So lymphocytes trigger antibody production. This is adaptive immunity, which specifically bind to epitopes via complementary paratopes. So moving on, so these are cytokines and cytokine receptors. So during sa atong immunity processing when it comes to ma-infect certain foreign material, cytokines are just small proteins that are crucial in controlling the growth and activity of other immune system such immune system cells and blood cells. So when they are released, they signal the immune system to do its job. So also they affect the growth of all blood cells and other cells that helps the body's immune and inflammation responses. So these are cytokines are important roles in both innate and adaptive immune systems, which are major means by which immune cells communicate. So majorly, the function of cytokines is to regulate inflammation and play a vital role in regulating the immune response in health and diseases. So cytokines can control connective fibroblasts, endothelial, mast, lymphocytes, macrophage, and granulocytes. So moving on, aning nga table, this is the list of all the cytokines and the principal source, kung aha siya gikan, and the possible and its functions as well. So I think ang interleukin ma-mention na earlier, I mean later, sa atong mga figures. So dito niyo makita ang mga more in detail versions ani nila, which is during the cellular signaling process kung kano sila mogawas when it comes to interact na dayon when it comes to during the signaling, and then it comes to the byproduct, which is to defend the host from the certain foreign material and to release a specific needed tawag ni immune response nga haom siya sa foreign material na ilahang gi-fight with certain foreign invader, if nanabay or nanabatay memory cells against ani, if wala unsay buhaton sunod, so somewhat na ang ginabuhat. So ani nga part, so next so other chemical defense. So diri for example the mucus, so this is the mucous membranes lining the digestive and respiratory tract of vertebrates and contain parasiticidal substances such as IgA and lysosome. So we all know IgA is a class antibody. It's part of the adaptive immune system. And then also IgA can cross cellular barriers easily, and it's a protective agent in mucous and intestinal epithelium. Somewhat if na parasite na nakasulod within our intestinal epithelium, epithelium or digestive system. So IgA and lysosome would be the first line of defense since sila man ang present dito sa membrane lining sa atong digestive and respiratory tracts. Also, IgA is present in saliva and sweat. And then lysosome as well is an enzyme that attacks the cell of many bacteria. So this enzyme, like the lysosome, is naturally occurring found in bodily secretions such as tears, saliva, and milk. So it functions as a antimicrobial agent which cleaving the peptidoglycan component of bacterial cell walls, which leads to cell death. So moving on to interferons. So interferons are proteins that are part of your natural defense. So basically, interferons are the very first gatels in my body na you need to make that you are needed to prepare immunity or immune system for the said foreign material na nakasulod. So here, so moreover, interferons tell your immune system that germs or cancer cells are in your body, and they trigger a killer immune cells to fight those invaders. So interferons got their name because they interfere with viruses and keep them from multiplying. So diri siya, interferons are type of cytokines released by a variety of eukaryotic cells in response to invasion of intracellular parasites, which includes viruses and other stimuli. So na-describe ni si interferon as an antiviral factor because they interfere the replication in mammalian cells. And then they secreted also from infective cells and activate immune response that promotes not only cytokine production but also natural killer cell functions and antigen presentations. So moving on for the tumor necrosis factor, this is mainly they are produced mainly by macrophage but also by activated T cells and natural killer cells. So this is the major mediator for inflammation, and iyang insufficient presence could lead, which causes fever. Because a fever in mammals is one of the most common symptoms of infection and is a fundamental defense mechanism because high body temperature may destabilize certain viruses and bacteria as well. So another cellular defense would be phagocytosis. So by definition, phagocytosis is a process of engulfment of invading particle with an invagination of phagocyte cell membrane. So here, this is an example of non-self recognition. So it occurs in all metazoa, and its feeding mechanism in many single cell organisms. So kani siya, if the foreign material is then micrometer plus more or mas more than 10 micro-m, dili na siya ma-engulf. Dapat na siya i-encapsulate. So the following are um, such as monocytes, PMNS or granulocyte mast cells, lymphocytes can do phagocytosis. So here, the figure, you see the lineage of some cells active in immune response. These cells, well, as red blood cells and other blood cells, are derived from multiple potential stem cells in the bone marrow. So makita na to iyahang different types of cells after sa pinaka first multipotent stem cells become myeloid stem cells, lymphocyte stem cells, and then it undergoes certain processes to be macrophage precursor, it becomes monocyte, and then macrophage na jud, and then is a B cell precursor, become B cells, and it becomes plasma memory cell. And so lymphoid stem cells, it becomes natural killer precursor, and then it becomes natural killer cells, and then it becomes activated natural killer cell. And then lastly is kaning isa, it becomes T cell precursor, it becomes T cell of course, and it becomes helper T cell and cytotoxic T cell. So muna siya ang lineage of some cell actives in immune response. So moving on sa monocytes, so they arise from the bone marrow and give rise to molecular phagocyte system. PNMS or the polymolecular leukocyte composed of neutrophils, eosinophils, and basophils. So there are types of white blood cells. Mast cells then is basophil like cells found in dermis and other tissues. Lymphocyte is crucial and adaptive immune response composed of T and B cells. So lymphocytes are the cells that circulate in your blood and part of the immune system. And also lastly, the natural killer cells. So these are lymphocyte-like cells that can kill virus-infected and tumor cells in the absence of antibody. So these are the these are the type of cells that can phagocyte foreign materials. Moreover, moving on to adaptive immunity response of vertebrates. So there are two, cellular and humoral. So adaptive immune response for starters is stimulated by a specific foreign substance called an antigen, and circularly an antigen is a substance that will stimulate an immune response. So the special system of non-self-recognition possessed by vertebrates results in increased resistance to specific foreign substance or invaders on repeated exposures. So for the cellular immunity, it's associated with cell surfaces, so it occurs inside infected cells, and it's mediated by T lymphocytes. And the humoral immunity, also called antibody-mediated immunity, based on antibodies, both on cell surfaces and dissolved in blood and lymph. So here, the humoral immunity protects against extracellular pathogens and also toxin, and the cell-mediated immunity protects against intracellular pathogens. So before differentiating humoral response and organic cellular immunity response, so there's our function of antibody in host defense. There are four: opsonization, neutralization, activation of complement, antibody-dependent cell-mediated cytotoxicity, ADCC. So here, opsonization is an immune process which uses opsonins to tag foreign pathogens for elimination by phagocytes. So opsonin, such an antibody, this is a negatively charged cell wall, so the pathogen and phagocytes repel each other. So dili daw, without the opsonin, dili daw mabuhat ang job sa phagocyte to engulf the foreign material, since opsonins are used to tag foreign pathogens so that ma-recognize siya sa phagocytes. Next is neutralization. So this is composed of IgG and IgM antibodies which neutralize toxins that are secreted by bacteria and prevent toxins molecules from binding to their target cells. Also, these antibodies can bind to the envelope of viruses and prevent the virus from attaching and penetrating the host cells. Next is activation of complement. So this is the ability of the antibodies and phagocytic cells to clear pathogens from an organism. So here, once it activated directly by pathogens or indirectly by pathogen body antibody bound antibody, leading to a cascade of reaction that occurs on the surface of pathogens and generates active components with virus effector functions. And then lastly is ADCC. This is an important mechanism against parasites since this antibody bound to the surface of an invader may trigger contact killing of invader by host cells. So this is the generation of humoral response. So this is just the totality or the components of humoral response such as the lymphokine activated killer cells, the cytotoxic cells. The IgG, IgM, IgA, and IgE came from B cells and the activated eosinophils. So humoral immunity is also called antibody-mediated immunity with assistance of helper T cells and B cells will differentiate into plasma B cell that can produce antibodies against a specific antigens. So as you can see, B cells that produce antibodies against specific antigens. So the humoral immune system deals with antigens from pathogens that are free circulating or outside the infected cells. So diri makita na to is that this is the major pathways involved in the immune response to parasitic infections, which is mediated by cytokines. So you already know what's the major function of cytokines. So ang solid arrows indicate positive signals and the broken arrows indicate inhibitory signals. So broken lines without arrows indicate the path of cellular activation. So di mag-ila-ila mo ni lymphocyte, the immunoglobulin, the interleukin, the TNF, the TNF or the tumor necrosis factor, the Th1 helper, CD4 and CD8 that stimulate cell-mediated response, the Th2 helper, stimulate humoral response, and RN1 reactive nitrogen intermediates, and so on and so forth. So I'll be trying to discuss this more on detail, but I'm going to check if it's really a part of sa atong syllabus since we're on focusing sa atong major topic which is parasitology. So nanon siya sa more paano siya cell bio and immune part na gid kaayo. So ako pang i-decide kung sa i-include ba gid na ako. Moving on. So this figure shows kaning sa inyong left shows the how macrophages, how immune system works. So dire, this is the example of a humoral immune response. So dire makita na to, follow lang ni 1, 2, 3, 4, 5, 6 for the cascade effects. So dire makita na to that the macrophage consumes the antigen. This is the viral antigen, and then we consume siya and digest. It displays epitope on its surface along with class 2 MHC protein and secrete interleukin IL-1 muna siya. So diri balik ta aning type of katong epitope na akong mentioned earlier, makita ninyo na ni-attach antigen sa epitope of the said antigen. So once it attach, ma-recognize nila ang say needed, so gi-release na din nila sa IL-1. So kani si the IL-1 or the secretes, this IL-1 or interleukin was secreted once the antigen attached to the epitope, and then T helper cell, which is stimulated by IL-1, recognizes epitope, and class 2 protein on macrophage is activated, then ye secrete and see IL-2, 4, 5, and 6. So these epitopes are recognized by the antigen-specific Th cells. So mo nang na-secrete sila dayon. And then, so they stimulate the specific B cell to proliferate and differentiate, and then it multiples rapidly, as you can see here, kani. And then it produces many plasma cells, which secrete large quantities of antibody for a period of time and then die. So here, Pag abot sa number 3, you can see the T helper cell then activates B cell. Kani siya, T helper cell activates the B cell to proliferate and differentiate large quantities. So, once na-activate na si B cell, it finally produces kani si sinayad B cell na-activate siya by the help of the T cell receptor. Pag activate na din ni B cell, since it carries the antigen and faster protein on its surfaces, ay makita ninyo na siya kani siya o kana, which is it carries the antigen na. And then once activated, some of the cell becomes plasma cells, this one, and others became e-memory cell for future use in case mobalik ni siya nga viral antigen. So this plasma, plasma cell antibody produced by plasma cells tend to bind to antigen and stimulates macrophages to consume antigen o opsonization. So mao ni siya. So natag naman siya by the natag sila sa opsin, which triggers opsonization. So then katuding mga phagocytes will help to consume the antigen since already tagged naman siya as foreign para dili na siya maka-invade sa host. So that's how humoral immune response works. So it's 1, 2, 3, 4, 5, 6, but if go pagid ani napadunay certain chemicals and then present, if somewhat mo zoom ka what's happening yun napadun na siya. So kanina siya is a quick overview of what humoral immune response looks like. So next is katong isa, cellular immunity or cell-mediated response. So many immune response involve little, if any, antibody type and depend on the action of the cells only. So in cell-mediated immunity, the epitope of the antigen is also presented by macrophages as well. And then also cellular immunity is a protective immune process that involves the activation of phagocytes, antigen synthesized cytotoxic T cells, and the release of cytokines and chemokines in response to antigen. So this type of immunity is effective against cells infected with viruses, intercellular bacteria, fungi, and protozoans and cancerous cells. So it also mediates transplant rejections. So here, there are 4 types of cellular immunity. Delayed type hypersensitivity, cytolytic T lymphocyte, natural killer cell responses, and immediate hypersensitivity. So to know a bit more about these types of cellular immunity, so muna sila. So the delayed type hypersensitivity, this leads to inflammation. Cytotoxic T lymphocyte responses. So this is important for organ transplant rejection and viral infections. And NC cell response or natural killer cell responses, there are large and granular lymphocytes that express neither T or B markers on their surface. So kani sila is very important in organ transplantation and viral infection, but it tends to occur earlier than the CTL responses. So pinaka-first pa siya mo gawas than the CTL. And then the immediate hypersensitivity, this is an allergic reaction provoked by re-exposure to a specific type of antigen referred to as an allergen. So this is probably by exposure due to ingestion, inhalation, or injection, or possibly direct contact. So that's it for the cellular cell-mediated immunity or cellular immunity. So for the next meeting, I'll be continuing to discuss still the basic principles for the ANI and the ANI, and I think I can finish it good as well, and I can send na the PowerPoints as promised. But isend lang nako till katong isa katong second lecture. So isend ako is 1 and 2. And then once mahuman ni by next meeting, ako na din siya isend, and that would be the coverage for prelims. So I hope everyone is studying by then. Also, I hope by next meeting, I probably I don't know what is it next Saturday. I hope I can receive the tawag ni outline for your reports atong sa mga tawag ni sa articles given para maka-start na ta dayon after prelims, since I'm not sure kung sa ito na ko giingon ang start, but I think kuanto ang date na kung na-mention is murag examination date, so move na to siya. So after sa exam na siya mag-start, so prior like anang a week, the Wednesday after the examination week, we will start na. So I hope group 1 to 4 would prepare na already and also give me the outline so as soon as possible. Since we're going to do this as fast as we can sa reporting, and don't really pressure yourself if kanang into learning the tawagan ni katong mga detailed kayo, not necessarily. I wanted you to just understand the article itself lang. So unsa jud inyong nasabtan lang, and if you have time yun sad to check unsay buot nilang pasabot, that would be great since lang may ma-learn new things na sad. So that would be possibly additional points but if ma-deliver na ninyo by during the reporting. So that's it. Thank you so much for listening, and also please send me a message once you're done. Hopefully we will do synchronous session by next meeting na gid ni. The last synchronous session prior sa atong exam. So probably next Wednesday will be the last synchronous for the prelims exam. So if you have questions with regards to the, I don't know, baka anything, or like unsay akong akong basihan sa akong exam. So yes, mo to siya. Thank you so much and have a great weekend. Thank you so much, guys.